Hello, thank you all so much for joining us for this roundtable discussion about drug repurposing and the ACD-funded fellowship studies that are collaborating toward a common long-term goal of finding precision medicine treatments for creatine transporter deficiency via drug repurposing. I've been fascinated as I have learned more about drug repurposing and its potential for treating CTD over the past year, and I'm hopeful that today's conversation will help our community to have a better grasp on what drug repurposing entails and how it might better be able to help our loved ones affected by creatine deficiencies. So first, let's all introduce ourselves and our connections to ACD. I am Erin Coller. I'm an ACD board member and mother to my son Cadman, who is 6 years old with CTD. Jonathan, go ahead.
Hi, I'm John Schlebach. I'm an assistant professor of chemistry at Indiana University in Bloomington. We've been studying in my group creatine transporter deficiencies for about 5 years. Yeah, I mean, it's a project that's sort of close to my heart because I have 3 autistic nephews and I've been interested in this sort of spectrum disorders for a while. So.
Great, Peter.
My name is Peter Xerachilius. I'm an investigator at BC Children's Hospital. I've been working on these type of studies for the last 3-4 years. My expertise is more focused on drugs in general and repurposing drugs, and for these kind of rare diseases, particularly CTD, and hope that I can make a difference in finding a safe and effective drug for this purpose.
That's wonderful. Thank you. Hi, Sangeetha.
Hi, everyone. Good morning. My name is Sangeetha Iyer. I'm a pharmacologist by training. I've worked in the field of rare diseases for a couple years now, and I've been affiliated with the ACD for over 2 years as their scientific advisor.
Thank you all so much for being here. So I'll get our conversation started with our first question. John, I was thinking you could answer this one, get to kick us off. If you could please explain drug repurposing.
Yeah, so basically, you know, this is a sort of type of drug discovery, right? You're using the tools of drug discovery, and you're hoping you're going to find a drug that was developed for another purpose that's going to work for a new disorder. So it's important to keep in mind when you develop a drug for a specific purpose, right? You're targeting, one of many proteins, right? So there's a lot of proteins in your body that interact with sort of a common set of small molecules like creatine. And so we're hoping we're going to find, a bioactive molecule that's already approved for one treatment that just so happens to also help with creatine transport deficiency.
Okay. And, I think the next question you can jump in on too. What makes CTD a good potential candidate for drug repurposing?
Yeah, I think this is one of the things that drew me to this as a tractable problem. Mostly because creatine is a super abundant metabolite. And there are a lot of other molecules in your body that look like creatine, which basically means a lot of the molecules that are going to interact, with other drug targets are also likely to interact with creatine transporters. It's just that nobody may have looked for this in the past, right? So this is one of the reasons why I think that this is a promising approach, just because, again, there should be lots of other drugs and small molecules that interact with creatine transporter we just don't know about. So I don't know if that answers the question, but that's the way I see it.
Yes, that is very helpful and hopeful too. Sangeetha, I think this leads into something I was hoping you could share with everyone, which is, can you tell us a bit about your past experience with drug repurposing for a rare disease?
Sure. So a couple of years ago, I was working for this company called Perlara, and we were working on a repurposable drug screen for this rare disease called phosphomonomutase 2 deficiency. And we were conducting the screen using simple model organisms as well as patient fibroblasts. So when we conducted the drug screen using our simple model organism platform, we identified a few molecules that were able to restore function of a critical protein that has lost function in this particular disorder, PMM2. And when we did some follow-up research on that particular molecule and tested it in patient fibroblasts, we found that it was indeed able to restore function, not just in the engineered model systems, but also in the patient fibroblasts. So for context, patient fibroblasts are cells that are directly derived from, say, skin biopsies from a patient suffering from a given disorder. And in those particular cells, this particular molecule was able to rescue this enzyme defect produced by loss of the protein. So that led to additional follow-up studies on the patient fibroblasts and the patient in question by a clinician at the Mayo Institute, which then led to an N of 1 clinical trial around that molecule.
Thank you so much. Yeah, that's we're hopeful to be able to head down a similar path. So we're very, very fortunate to have your guidance helping push things along here at ACD for creatine deficiencies. And I would like to ask a question that is open to anyone to answer. What are some of the biggest challenges that we face with identifying a drug that is suitable to treat CTD?
I think Peter might be a good person to start on this.
So the biggest challenge is, one is we're not just looking for, well, definitely looking for a repurposable drug, but a repurposable drug that is obviously safe, can be taken, orally, can be taken every day, and that is effective. And what I mean by effective is it does what it's supposed to be doing, but also at a low dose, at a conceivable dose that a human can actually take. And also that the drug needs to get into the brain, because that's where the disease is affecting these patients. So these are the criteria in which the drug needs to have these properties. And one of the biggest challenges is to find a drug that has all these qualities. So the main goal, at least the basic goal, is to take a step-by-step approach where you would find a drug, repurposable drug, that has at least one quality. Then you can find similar drugs that may have then one extra quality. For example, it's more effective, or it's safer, or it's better at getting into the brain. So that's the sort of thing that's the biggest challenge, I would say, for drug repurposing.
Yeah, I'll add a little bit to that. So, I mean, from my perspective, so I train in pharmacology too, and when you see most of the drug discovery processes, right, you cast this really large web, right? Like the number of shapes and sizes of molecules that you can make that are drug-like is virtually infinite. And the problem is there's only a few thousand that have gone through the lengthy process of clinical trials and have come out successful, right? So we're sort of, we're not able to cast that huge wide web when it comes to discovery. So you're sort of trying to thread a needle here. But the upside is these molecules, again, they are approved. We know they're generally safe, and we know that they're generally bioavailable, right? So they've already passed, checked those boxes for other diseases, right? So that's sort of the way I see it. And for this reason, we're also pursuing sort of larger screens of molecules that aren't current drugs, but we're prioritizing the ones that already are drugs, just because we don't have to go through clinical trials for those.
Thank you. And I think this brings us to the studies that you're both working on. So as we all know, that you both have studies you're involved with that have been funded through ACD's fellowship program and have about 1 year under your belts, approximately. And we are waiting, find out soon about announcements for the second rounds of funding for these studies, and would love for you to help explain today some of the basic goals and focus of these studies and how you're both collaborating together.
So the two Jonathan is he has a sophisticated system where he can actually quantify, and he can discuss that. He can actually quantify how much of this transporter, this protein, what went wrong with it in terms of, is it stuck inside the cell? How much of it is actually inside? And is it actually at the surface? And that's where it needs to be for it to function. My studies are focused on trying to find, once the transporter, the protein, is at the surface, or in some cases, these mutations just causes a dysfunctional protein at the surface. My studies focus on trying to restore the function of these transporters at the surface. So our collaboration together is two different ways of approaching this subject and trying to find a repurposable drug for different different types of mutations that have affected the protein.
Yeah, I think, yeah, just for context too, Peter, as he mentioned, right, has had a successful career already in terms of drug discovery, and that's sort of his wheelhouse. And I think what my lab, we generally have about 10 to 12 people in my lab, and we study a lot of different diseases, but they all have sort of similar genetic basis to creatine transporter deficiency. So we're trying to take some of the tools that we've developed and perspectives we developed from other diseases and translate those into creatine transporter disease. And again, in terms of the nuts and bolts, right? Like Peter said, they're focused on function. We're focused on protein expression and localization. In terms of the computational tools, Peter is, sort of moving ahead with traditional tools. We're trying to develop some secondary tools that'll help us sort of zero in our search on the molecules that might be somewhat different or complementary. And so that's sort of the way I see it, right? And I mean, just from a general perspective, right? 4 heads are better than 2 between Sylvia and Peter and me and Charles, my associate. So I think that's the thing. When we meet, we talk, we come up with new ideas, and it's been really beneficial, I think, from our end. And I'm excited about where it's going.
So, Jonathan and Peter, if maybe if you could add some more color to this aspect that I have been thinking about, which is that based on your study of mutations for affecting the creatine transporter, they're not all the same, right? In that some of these mutations may affect the function, some of these mutations may affect the expression itself, right? So the kind of study that you both are doing helps you find drugs that can rescue the defect at both levels, right? Would that be fair to say, fair assessment of your work?
Yes, very much so, yeah.
Correct. And there is precedent for this sort of approach with other diseases, most, namely cystic fibrosis. I think what we've learned about creatine transporter deficiency in terms of the molecular basis of the disease, we can learn a lot of lessons from cystic fibrosis and other similar diseases about how you would go about finding drugs and targeting them to specific mutations. There's been incredible success in that realm, and I think we can definitely benefit from an understanding of how that's all played out.
Yeah, and I think that cystic fibrosis is also an interesting case because in cystic fibrosis, weren't they able to find a drug that actually acted across types of mutations that were similarly disrupting the function of that particular protein, right?
Yeah, so basically, by treating expression and function separately and looking for molecules that do different things the way that Peter and I are doing, they've approved several molecules, and what they currently do for most genotypes is for most patients, they basically give you a combination of molecules, some that increase function, some that increase expression. But if you don't have an understanding of what the mutations do, then it's hard to interpret the effects of the molecules you're looking for. So I think that's the real lesson from cystic fibrosis, is just realizing the mutations are not all working in the same way. And if you don't pay attention to that, you might miss something interesting, right? And then B, when it comes time to use your molecules, you have to think about what the mutations do and what the molecules do and match them up, right? So that's sort of what I'm alluding to in terms of what Peter's doing, what we're doing are complementary for this reason, because we're trying to understand what the mutations do and what the molecules do, and then, you know, match them up.
Yeah, that's a really good segue into why it's important for us to understand these different types of patient mutations and how they might potentially affect function, right? And so we always think about this, which is what's the best way to sort of identify all of the mutations that are out there that are presenting in patients. And that's one of the reasons that we encourage people in the community to also deposit their fibroblast. It's a really simple thing to do. You have to take a skin biopsy and then send that over to Corial, and then that gets converted into fibroblasts that match and have the same genotype that that you do, that your loved member does. And that enables us to actually distribute these fibroblasts, have researchers study them, and understand how your particular mutation might be affecting the function of the creatine transporter or other protein.
That is such an important point. And I think that I love that you brought up the example of cystic fibrosis. It's so helpful for all of us to see examples like that and how we can learn from them, and gives us some hope for what we might be able to do for CTD as well. And I would love to dig in a little bit more about your specific studies. So Peter,
can I add one note? Sorry.
Yeah, absolutely.
On this note, just since this is about ACD, I should note that the success in cystic fibrosis was 100% due to the Cystic Fibrosis Foundation collaborating with both industry and academia over an extended period of time. So this is one reason why the ACD is super important, because it brings people like Peter and me together to work together towards finding things, right? You really need a foundation like ACD to do that. So sorry to interrupt. I just thought I'd point that out. That was a critical part of the success there.
I appreciate that. Thank you so much. And Peter, can you tell us about some of the key findings in your study so far?
Absolutely. So it's basically what we alluded to just now about talking about how not all variants are created equal. So the main goal of the study was, the initial part was to find to understand what the diagnosis for each of these variants are. What I mean by that is it, is it dysfunctional? How dysfunctional is it? And trying to understand these problems. So the first key finding was we tested various mutations or variants and tried to figure out what were these consequences and quantifying them. So we found that there's some mutations variants that were partially defective, some that were completely defective, and that's sort of what we expected. But then we did a screen on millions of drugs, including the FDA-approved ones, also some natural products. And we found that some initial leads or hits that could restore partially the function of the transporter, which is a great start. Usually you will not find a drug just on one screen and then the study's over. So it's going to take a step-by-step approach, trying to find initially drugs that work slightly, and then we can work on that to find similar drugs that may have same structural features, to find them that are possibly more effective, and definitely in the realm of repurposable drugs. So we found some that worked and that could increase the function at the surface. So now we're the next steps are to find similar ones that are more effective at a lower dose.
Okay, that is good progress within the first year of your study, for sure. And Sangeetha mentioned earlier about the importance of our community submitting those patient samples to the Corial Biobank, the blood and skin biopsy samples. We have a bunch of information on the ACD website at creatineinfo.org about how to do that for our families in our community. And I would love if you can share with us how that biobank is playing a role in your study.
Yeah, so it's really important to have what we call a model, and an accurate model of what a patient or the disease affecting those cells. So we use engineered cells initially to do the screen, but ultimately we need to understand how the drugs work in a patient cell. And these Coral Biobank gives us the ability to test these drugs with these fibroblasts. And it's once we've identified a drug, repurposable drug that is effective and it works well in our engineered cells, it's really important to test these in using these fibroblasts and try to see how effective they are in those cell line. And then once that works out, then we can safely say that the drug is promising and should ideally work in a patient.
Okay, and so would the next step be, once that step is complete, would involve an N of 1 trial potentially? Is that correct?
Yes, that's potentially the next steps. Yeah.
Sangeetha, can you jump in here and explain to us what an N of 1 trial is and what is the path for repurposable drugs to go from N of 1 to an expanded clinical trial with more than one participant?
Sure, I can share with you what I know based on my experience with PMM2, with the caveat that the path may not be the same for all diseases, right? Every disease is unique and treated somewhat differently based on the burden of proof of evidence that we have at the time. But at least in my N of 1, quite literally, experience around this trial. So an N of 1 trial involves the use of a safe drug for one single patient. And there is a regulatory process that has been outlined by the FDA that you have to follow in order to initiate something like this. It does involve getting essentially regulatory approval from the FDA to enable dosing an individual, just a single individual, on compassionate grounds or other grounds to really test this molecule, test a particular drug in a single patient. There is usually a clinician that is involved in determining whether such a study can take place. The clinician is also responsible for monitoring whether such a drug is going to be safe in the patient, and if the drug is efficacious, then what might the markers of efficacy be. So all of this information has to be compiled in a package following any testing in patient fibroblasts and submitted to the FDA. FDA reviews such a package, and if they find that all of the information that they require is there and is complete, and they're convinced that the case for an N of 1 trial is made, then they provide permission to the clinician to go ahead with the trial. Now, the clinician is usually responsible for determining the shape and format of the trial, right? So what's the dosing that they will follow? Will they do any baseline, that is no drug measurements? Following drug dosing, what kind of measurements they will make? And for how long they will treat the patient with the drug, and identify metrics of success or failure of the drug. So they initiate the trial along this format, and if they are successful in finding the sort of information that will convince the FDA that the drug is safe and efficacious, then they submit a follow-on package to the FDA outlining what an expanded clinical trial might look like. And then the FDA reviews that package, and if it is convinced, then can provide approval to initiate such an expanded trial. Now, going back to some of the specifics that I've been aware of, for PMM2, like I mentioned at the start of our conversation, the N of 1 trial was initiated by a clinician at the Mayo Clinic, and they provided data from the patient fibroblast indicating that the drug rescued the protein defect. They were lucky because there was a lot of available information about the drug in terms of its safety itself that was publicly available, because the drug had been approved elsewhere for over 20 years. So there was a ton of safety data that they were able to rely on. The challenge for them was in presenting to the FDA what they would measure in terms of improvement in the presence of a drug. So what's a biomarker that will speak to the efficacy of the drug? The reason this is important is because oftentimes for a rare disease, because the clinical presentation of the disease is multifaceted, right? So you have motor difficulties, you may have some cognitive difficulties, you may not see correction in all of these immediately. And it can be considered unethical to let a trial go on for a long period of time until you actually see correction in these gross effects. For this reason, the FDA likes to see a clinical biomarker. That is something that is measurable, quantifiably measurable in the blood or some other system that can speak to the efficacy of the drug before you can see correction of gross effects. So because this was a rare disease and there was not a lot of information for what a biomarker might be, there had been no clinical trial for this disease done before. There was some amount of work that the clinician and their team had to do to propose what a biomarker would be, and to conduct the right kind of studies to show that this is something that changes when the disease is course correcting. So that was the type of information that they were able to gather during the course of the N of 1. And then they published on it, and it became, I think it is on the process of becoming a validated biomarker for this particular disease, which gives them that body of work to build on when they go into an expanded clinical trial. The other thing that I want to mention is something that Peter spoke about early in the discovery process. The other reason why we were able to move to an N of 1 trial for PMM2 is because the drug was orally bioavailable. There were manufactured dosage forms that had to be recompounded. So in other words, what you're saying is, let's say there's a drug out there that's just made for an adult population, right? An adult dosage form is not something that you might be able to easily administer to a pediatric population. So you may have to remanufacture or remake the drug in a more suitable manner, a manner that's more suitable for dosing to pediatric population. Because you're changing the dosing format, some additional tests may also be required to determine that the drug compound is actually present in this reformulated material. So there are, apart from the efficacy considerations, safety considerations of the drug, there are also these manufacturing considerations, pharmaceutical formulation considerations of the drug that the FDA looks at. So that's another piece of the puzzle that is required that you have to bring together when you're thinking about an N of 1 trial. So just because I know that our community is very, very interested in seeing an N of 1 trial happen, as are we, but I do want to point out that even after testing of the drug in patient fibroblasts, there is still some amount of work that we would have to do, that anybody involved in this process would have to do, to check all of these boxes that the FDA has outlined for the purposes of a safe and successful clinical trial that we will have to go through in order to initiate an N of 1. Does that answer your question, Erin?
Yes, and more. Now I have 5 more questions. But one of them is that, when you're talking about biomarkers and having all of this data and information about what would be meaningful outcomes for our patients, would you say that our registry and the patient meaningful outcomes survey in the registry, the PCORI grant, and the work that we'll be doing through that, is that all going to help lead us in the right direction to get us to the point that we would be able to have an N of 1 trial happen?
Absolutely. I think the registry is particularly important because through the registry, we are able to collect the sheer diversity of the patient population that's out there, right? So as much information as can be uploaded by the families regarding age of diagnosis, regarding the patient mutation, regarding the clinical presentation of the disease in their loved ones, right? So all of this helps us build a very accurate clinical picture of the disease across different ages. So one, for that very reason, the registry helps us capture the diversity of this disease in the community. The core outcomes work that we are hoping to do through the PCORI grant, that will help us capture what are the outcomes that are most meaningful for families to see correction in. Are they cognitive? Are the biggest challenges motor related? Are the biggest challenges intellectual disability or behavior related? These are things that we as a community should be knowledgeable about, because this is the kind of data package that would help us tie together what's the type of correction we want to see when we dose with a drug. So it helps us build together this picture of if we were to see correction, what would be the meaningful endpoint? How might we be able to quantify it? And all of this groundwork, if we can do it before we have to initiate any clinical trial, it'll certainly help us help the clinician, help the researchers working on this disease put together a more complete package in front of the FDA. They'll be able to refer to some of this body of work and say, we know the presentation of the disease. We know that these are the things that the community wants corrected. And these are the ways that people have said that they can be measured. And that will help us putting a more more robust package in front of a regulatory body.
I think that just helps so much for us as parents to connect the dots and really understand why the fact that we have this opportunity to play a lead role in the research and to take steps within our power to help get us to these clinical trials that we so desperately want. So I guess my call to action and kind of plea with our community is just stick with us and keep watching for all of our requests for more surveys or fundraising efforts, and just knowing that this is why we're doing all of that, and supporting studies like this and doing our part to gather all the correct data and to do everything that we need to do to make it possible for all of this to move forward. Which, brings me to our next question, which is just about both of these studies. What are the next steps? And as I mentioned, we're expecting an announcement soon about the next round of funding, and would love to hear what is proposed for the next phase in each of your studies.
You want to start, Peter?
Yes. So for me, it's first of all, the collaboration with Jonathan is a key in terms of once we do our screening of the drugs, we'll test these drugs for different purposes. Like we said before about testing drugs on the transporter that has been sort of stuck inside the cell, in particularly in cases where the protein is dysfunctional at the surface. So we'll be doing some screening on trying to find drugs for specific purposes. We'll also take in the information we have so far with the drugs that have been working, try to find some common structural features, and try to find similar drugs that have been FDA approved, and test them. And then the main goal is to obviously to find a effective drug that gets into the brain and is effective in those cells, and then test them in the coral fibroblasts patient cells, and try to see if it works in that model. And then from that point, then we can continue on, because it's a multifactorial with different variants. It's key to test further all the variants and all the and all the variants, and diagnose these variants, and then test all the drugs, group the variants, and then test all the drugs on these different type of issues. And again, to test them and coral. So I really encourage everyone to, if they have the opportunity, to deposit their fibroblast cells at the Coral Institute.
Yep.
Thank you.
On our end of things, so Peter has sort of moved forward with the first round of testing using sort of traditional screening approaches. On our end, we've spent a little bit of time developing some sort of complementary tools to identify different classes of molecules that might also be useful. So what we're doing right now is basically screening up or setting up another search, and we're going to try to identify a new set of molecules that we think are promising, compare them to the original set that Peter has tested. We're going to try to look at them side by side and try to understand sort of what makes a good molecule, what makes a bad molecule, and try to use that information to identify sort of, you know, other promising candidates. So that's what we have sort of on our mind is, you know, A, you know, taking our new tools and using them, and B, trying to learn from, you know, Peter's current experiments, sort of what works and what doesn't and why. And I think, you know, we're at this sort of stage, I think, you know, we have a lot of tools and a lot of data that are going to point us in some new directions. So I'm excited to see where that goes. We also, on the experimental side, we're setting up some new screening tools so that we can search molecules more efficiently, you know, in the laboratory, in addition to sort of computationally in the computer. So that's sort of what's on our agenda.
Excellent. You know, I think we actually covered all of my questions. So unless there's anything that anyone would like to add, we can wrap this video up.
I have a question. Maybe it's not relevant, but in terms of can families, in terms of finding, Sangeetha, you've been saying about biomarkers, could families start to watch or take notes of how the patients are doing, and could that help clinicians to understand better what to look for and use that data or that for the clinical trials? Or is it more the clinician needs to look at that, or will it be the families will be helping the clinician to understand what those biomarkers could be?
Right. So I think it, typically what happens is that families, when they record improvements and the like, that becomes part of the qualitative narrative. But I think the FDA definitely places a lot more importance on quantitative or measurable changes, right? Now, and that's part of why we are doing this PCOD work as well, that I can envision a situation where families talk about changes that they are seeing. Maybe that is not something that we are able to capture in a quantitative manner right away, right now, because that's not on the radar of a clinician for something that is to be measured. So we are looking at new ways and looking to clinicians that are developing different methodologies for other disorders to see if there are more newer, novel quantitative methods out there for quantifying some of the challenges that are encountered by our community, right? And so if families are able to put those on our radar, either through the registry or through communications with ACD or their clinician, then I can envision a situation where we try to design something quantitative around it. it would need to be published and go through some version of a validation process before it becomes something that the FDA actually accepts.
Oh, fantastic. Okay.
That's the exact type of collaboration that we're all here for. So I love that we got to do a bit of that. So let's keep the conversation going offline for sure. And so I would love to just wrap things up with a couple reminders for our community watching this video. So we're doing this video right now at within the last few days of our Holiday Heroes campaign, a fundraising campaign, and I hope that this provided some insight into really where those dollars are going and why we have such lofty and ambitious goals this year, because of the next potential phases of studies like this, and what will really be involved with getting to clinical trials will take more and more funding and resources, and we're on the right path to get there, but we need the community's support to do it. So our community can support drug repurposing efforts to help get to clinical trials through donations to continue supporting these studies, and with Coriell Biobank patient sample submissions, and also with filling out those surveys in the patient registry as well, the creatine info registry. So please stay tuned for an announcement coming very soon about the next round of funding for ACD's fellowship awards. And can't thank you all enough for your time today. I learned a lot today. And so I am pretty confident that our community will as well. And I think this was really valuable. So I appreciate the discussion and all the information you all shared.
Thanks.
Thank you.
Thanks so much. Happy holidays, everyone.
Thank you. Happy holidays.
Happy holidays.
Bye.